Okay, team, today we begin our unit on genetics. And I just wanted to show you where the PowerPoint is. We are now in module 5. Again, I know it jumps around quite a bit, but here's the genetics PowerPoint. So if you want to open that up at any time, you can do so. And we'll do about half of the unit on genetics today. And we'll see how far we get. Okay, so who do we have to thank for this? Gregor Mendel is who we have to thank. Gregor Mendel is considered to be the father of Mendelian genetics. And so, I don't know, to me, he looks like a young David Letterman in this picture. I think it's the hair, maybe, in the glasses. At any rate, you probably are thinking Mendel must have been some hotshot scientist, right? But he was not actually a scientist at all. He was, in fact, a monk. He was from Austria, and he lived in a monastery. And turns out Mendel was a very curious man. He spent a great deal of time wondering about things. And his job at the monastery, if you know anything about a monastery, you know it's pretty much of a commune. There's not a lot of interaction with the outside world. And so they pray a lot, and of course they eat and they sleep, and then they all have chores. And Mendel's job was to be the gardener. So Mendel grew the vegetables for the monastery. Well, he got very curious about how certain traits were passed on from the parent plant to the offspring the next year when he would save seed and plant them. And so then he began conducting experiments to try to understand and figure out how this all worked. So that's kind of the history of Gregor Mendel. So here's the thing about genetics. If you don't take the time to learn the vocabulary, you will fail at genetics, because these are word problems, much like a math word problem. If you don't know what the words mean, then it's really hard to work the problem. And so what I'm going to tell you is that this slide is probably the most important slide in the entire slideshow, because it tells you what these different words mean. Okay, so let's take some time to go through this and try to understand what it is that we are doing. Okay. So first word that we have is this word that says genes, right? The best way to think about what a gene is, honestly, it's really a location on a chromosome. And that particular location is dealing with a specific trait in the organism. And so I mentioned this in an earlier slideshow or PowerPoint, but you know, if you ask a room full of students, you know, name some genes, usually I get hair color, eye color, skin tone. These are some of the very first things we notice about people when we first meet them. And so it's not surprising that these would be things that come up. The way that we can draw think about this, if I draw a pair of homologous chromosomes right beside each other like this, you know, here's a pair of chromosomes that contain the same genes. And so we could draw these little lines to represent the different locations where there are genes. And there are lots of different genes. Some genes are either or genes, and that makes them a little less complicated than something like hair color or eye color, right? Hair color, your hair is not either blonde or black, right? There's a lot of in between. But there are genes that are either or genes. For example, there's whether or not your earlobes are attached or unattached. So there's the earlobe gene. Some people's earlobes are attached. Some people's are unattached. It just depends on the individual. It's genetic, okay? There are lots of other genes. There's a gene for widow's peak. Y'all know what widow's peak is? It's when you have someone's hair, there's their eyes and their nose and their mouth. It's when their hair comes down like that, like Dracula. It's genetic. There again. There's the hairy knuckle gene. Maybe this is the hairy knuckle gene on the phalangeal digit of your fingers right here. You either have hair or you don't. I have hairy knuckles. Okay, it doesn't matter if you're male or female. So you get the idea. What makes this a pair of chromosomes is they have these same genes in the same order. And that's important to understand. Okay. What about alleles? Well, alleles are different versions of the same gene. Okay. So what does that mean? Well, here's a pair of chromosomes. Let's say this is the eye color gene. Alleles are a way to explain the different versions of that gene. So an example of an allele could be brown or blue. And those would be different versions of the same gene. The gene is still eye color. The gene hasn't changed, but now the expression of the gene or the versions of the gene can be different. Okay. Traditionally, we have used letters to represent alleles in order to work these problems. And so what that means is we can use, we'll get to that in a second. Okay. So let's think about something that Mendel did. Mendel looked at flower color in peas, okay? And he observed that some peas had red flowers and other peas had white flowers. The plants, he couldn't look at the plant and predict it until the plant flowered, but then some of the plants would exhibit these red flowers and some would exhibit white flowers. You could say those are different versions of the same gene, okay? And it turns out that sometimes these alleles can be dominant and sometimes they can be recessive. So dominant versus recessive. And so what we then do is we use letters to represent the alleles. And it turns out that in peas, red is dominant over white. That's just the rule for whatever reason. And so what we then do is we assign a letter for the red allele and a letter for the white allele. And we use an uppercase letter to define or represent the dominant allele. So red is dominant, so I'm going to use a big R to represent red as an allele, a version of the gene for flower color. Now, what are we going to use to represent white? Well, you might think a little w, a lowercase w, but because it's the same gene, you use the same letter. So not a little w, but a little r, like this. Okay, so now we have, this is called assigning alleles. And if you know that one allele is dominant over the other, you will use a capital letter for one and a lowercase letter for the other. The letter you use is irrelevant. I like to use the first letter of the dominant allele. That's why I used an R here. Makes no difference. You can use A, B, C, X, Y, Z as long as you use uppercase and lowercase letters, okay? So now let's think about some pea plants, okay? Let's think about what are some possibilities, right? The trait is controlled by a pair of chromosomes. There's the flower color gene. Okay. And so each individual gets two alleles, right? There's a spot for the alleles here and a spot here. And so what are some combinations of these alleles that any individual might have? Well, the individual could have two big R's, right? That's one possibility. The individual could have what? A big R and a little r. That's another possibility. Now, you might be tempted to say a third possibility is little r, big R. Don't switch these around and think it's different. It doesn't, think about it, if it's like this, big R and little r, it doesn't matter where the big R and the little r are, it just matters if they're there or not. So, you can switch these little r, big R, little r, it doesn't matter. It's only one possibility here. But the third possibility would be little r, little r. So we have these three different combinations of alleles, right? Big R, little r, little r, little r. Okay. With me so far? So now the next word in the vocabulary says homozygous. You should remember homo means the same. And so homozygous means having two of the same alleles. And so you look at these three possibilities here, big R, little r, and little r, little r. Which ones of these are homozygous? All right, well, big R has two of the same alleles. And in fact, we could call this individual homozygous dominant because it has two big R's. What about big R and little r? No, that's not two of the same. That's one of each, right? So the other one, actually, I know I don't have a lot of room here, and I apologize. Wish I had more room on the slide, but it's okay. So homozygous, big R, that would be homozygous dominant, or little r, that would be homozygous recessive. The point is that both of these individuals have two of the same alleles. The heterozygous individual, hetero means different. Heterozygous means having two different alleles. Well, how about a big R and a little r? Now, although you can say homozygous dominant and homozygous recessive, there's homozygous dominant and there's homozygous recessive. You can't say heterozygous dominant or heterozygous recessive. Heterozygous is just heterozygous, okay? Let all that sink in for a second. Now, we get to this word genotype. Genotype and phenotype are very important. The genotype is very simple. It's just what are the alleles, okay? So for example, big R would be an example of a genotype. I say, what's the genotype of this individual? You say big R. Little r would be another example of a genotype. This would be the homozygous recessive genotype. And then the last one would be big R, little r. This would be the heterozygous genotype. It's just, when I ask you for the genotype, look at the individual and say, what are the alleles? Okay, easy enough. The phenotype is the expression, the physical expression of those particular alleles of that individual. It's the expression of the genotype. And so when I ask you for a phenotype, ask yourself, what does it look like? Right? In this case, the gene is flower color. So what color are the flowers? So you could imagine if it's got two big R's, and big R is representing red, then you would say this flower's phenotype is red. Right? If it has a big R, oops, let's do little r first. If it has two little r's, well, that is the recessive allele that represents white. Since it has two little r's, these individuals would be white. That's their phenotype. Now, what about this one? Now, you might think, well, it's got one of each, but what does dominant do in this case? What does this say? Dominant masks recessive. What does a mask do? It covers it up, right? So although this little r is here, you would never even know it was, because the dominant allele masks this recessive allele. And so this individual is also red. Okay? So dominant, masked, recessive, unless you're told otherwise. That's pretty much it, y'all. That's like the vocab that you need to know for to do this kind of work, okay? And so if you know what these words mean, then what I'm going to suggest is you pause the video right here and you, let me clear the screen, okay? Pause the video, take a few minutes to just look over these words before we move on, because otherwise it's going to be a bit of a challenge in order to continue, okay? Okay, so now we're going to look at what Mendel did in his little experiment, okay? Like I said, Mendel got really curious about oops, where's my, I wonder what happened here. I'm missing a slide, unfortunately, it looks like. I may have to write this out. Let me see. Oh no, here it is. Oh, I don't know why it skipped to that slide. There we go. Okay, better. Okay, so, you know, like I said, it's a word problem. There's a bunch of words to know. And the words are important, right? We need to know what the words mean. So this is one of the first experiments that Mendel did. He observed that there were red flowered peas and white flowered peas, and he was very curious about this. And so what he decided to do was to take some pollen from one of the red flowered peas. He actually used a little paintbrush. He pulled the petals away. He took a paintbrush and he dabbed it in the pollen. And then he moved it over to the stigma, the female part of a white flowered pea. He's basically doing artificial pollination, right? He's doing sexual reproduction, but he's selecting a red flower and a white flower. And then he collected the seeds of the flower that he had pollinated. And then he was very curious what was going to happen if he crossed this red thing and this white thing together. Okay. So, you know, you might pause for a second and think, well, what would you think? If you didn't know anything about genetics, what would you think he was going to get? You know, he thought that half the seeds would be red flowered peas and half would be white flowered peas. He had never seen pink flowers and he had never seen both colors on the same plant. So he didn't think those were possibilities. He just figured, you know, if I collect 6 little peas out of this pod and I plant all 6, 3 of the plants are going to have red flowers and 3 are going to have white flowers. That's not what he got. And so let's do this as a genetics problem, which it is, okay, and take it step by step. The first thing you have to do is what we call assigning alleles. And so if red flowers are dominant over white, I like to pull that information out and say red, dom, white. So I don't have to keep going back and reading that. And then I'm going to assign a big R for red and a little r for white, just like we did before. So once you've assigned alleles, the problem will tell you what you're crossing together. This problem says cross a homozygous dominant individual with a homozygous recessive individual, and then determine the F1 and F2 genotypes and phenotypes. So we'll talk about what F1 and F2 mean in a second. Now, remember what homozygous means. Homozygous means 2 of the same alleles. Dominant means they're both big, and so one of the parents, one of the individuals is big R, right? It's homozygous and it's dominant. And I'm asking you to cross that with an individual that's homozygous and recessive. So 2 little r's. Okay. So that's, now these are the parents. Okay. Now, what the parents have to do in sexual, remember now we're doing sexual reproduction, right? And remember what the 2 events of sexual reproduction are: meiosis and fusion. So meiosis has to occur to form the gametes. And then fusion has to occur. And that's how you get to what we call the F1. The F1 is the first generation of offspring, and the F2 is the second generation. So we're going to carry this from one generation to the next. So really, every time you do a genetics problem, you're doing sexual reproduction, okay? If you just remember this, parents do meiosis to make the gametes fuse to form the offspring. We'll put offspring here just to remember, okay? Then you won't get lost. Now think about what's going to happen in meiosis. These letters, if you recall, are on chromosomes. Heck, we can even draw the chromosomes in if it makes you feel better, right? And what do chromosomes do in meiosis? They get pulled apart, correct? Now, in this case, these chromosomes get pulled apart and half the gametes get one and half get the other, but you will note that they're both the same letter. And so it doesn't really matter. The only gamete that this parent can make is a gamete that has a big R, right? Same thing here, the little r's get pulled apart. Half the gametes get this little r, half the gametes get this little r, but it's just a little r. Okay, so we have a big R and a little r. Big R from one parent, little r from the X. Now fusion just means you put the r's back together and there's your F1 generation. So there's only one gamete from this parent and one gamete from this parent. You put them together and you get this heterozygous individual, or this heterozygous genotype for the F1. The F stands for filial. Filial is like brotherly. So it's the first generation of brothers and sisters, essentially. And it turns out that they are all the same with respect to this trait. So what I want to ask you is a few different things. One of the things I want to ask you is, what is the genotype of the F1? Big R, little r. Remember, the genotype is just what are the alleles, okay? What is the phenotype of the F1? What does it look like? Well, dominant masks recessive, so all of these individuals are red, right? They all look the same. How many individuals does this represent? Now, this is an interesting question, right? You might think one, because I only wrote one out, but that's not correct. It represents a generation of individuals. I don't know, how many little seeds did you collect out of the pod? That's how many individuals there are. It just happens that they're all the same with respect to this trait. Okay, so did this make sense to Mendel? No, not really, because he's like, wait, I cross a red thing and a white thing together and everything comes out red. That doesn't make any sense, right? He kept thinking he maybe collected the wrong pod and he, tried it again. Same results over and over and over. Every time he got the same results. He got all red. Okay, so let's look at this. So here's what we did, right? We assigned alleles, big R and a little r. There's our parents. There's our gametes. Again, these get pulled apart, and the only thing this parent can contribute is a big R. The only thing this parent can contribute is a little r, okay? And then there's the F1 offspring. Now, in the original problem, I asked you for the F2. And so what we have to understand is, how do we go to the next generation? How do we do a second generation? Well, we do a second generation by doing what's called a self-cross of the F1. Let's look at what that means. What we're going to do is we're going to take this genotype right here and we're going to cross it with itself. It's called a self-cross. And it's kind of like incest, you're breeding a brother and a sister together, but in plants we can do that. It's okay. In animals, not so great, but when we're doing Mendelian genetics, we'll do it with animals too. It's just on paper, okay? So what I'm saying now is the parents now become the offspring. And so now what we're going to cross together is a big R, little r with a big R, little r, okay? So that's going to take us to the F2 generation. We're going to do a second cross. Okay, so now let's look at this. Here's going to our F2. Oops. Here's one parent. Here's the other. They both have the same genotype. Now, what's the step? Well, meiosis occurs, right, in these parents. And these R's get pulled apart. But look now, there's a choice, right? Half the gametes get the big R, and half the gametes get the little r. And then the same thing here. If the parents have the same genotype, you would expect the gametes are the same. Now, one thing to consider, it's not big R and little r, it's big R or little r. Does it make sense that the gamete has half the number of chromosomes that its parent had? That should make sense to you because that's what meiosis does. It reduces the number of chromosomes by 1/2. So if there's 2 R's in this parent, there's only 1 R in the gamete. It's going to be either a big R or a little r. Same thing here. Draw the box. What's the box? Well, there was this very clever French man named, we say Punnett in Western North Carolina. It's probably Punnett. It's spelled like this. In the French, they don't generally pronounce the last 2 or 3 letters of a word, which if you've ever studied French, you know. Okay, so here's how it works. You draw a box. This allows you to sort of merge the gametes back into the next generation. And the way it works is you draw a box, and what determines the shape of the box is how many gametes you have possibilities for each parent. Here we have 2, and here we have 2. And so I need 2 spaces across the top, 1, 2, and I need 2 spaces down the side. And then on the top, across the top, we put the gametes of one of the parents. And down the side, we put the gametes of the other parent. And then we fill in the boxes. And the way you fill in the boxes is you bring this R down, and you bring this R across, and so you get a big R and a big R. Here, you're going to bring the big R all the way across, and the little r down. Here, you're going to bring the big R down, and the little r across. If there's a big and a little, always put the dominant allele first. It's very important. And then this last box, look, we bring a little r over and a little r down. And this gives you the F2 generation, the second filial generation of offspring, which we created by crossing the F1. Okay. Now, I could say, okay, well, what about the genotypes and phenotypes? Now, the best way to express these ends up being as a ratio because we have choices now. And so I might say, how many different genotypes do you see here? Well, how many different genotypes? There's a big R, that's one. There's a big R, there's 2 of those. And then there's a little r. So the ratio then is 1 to 2 to 1, right? For every 1 big R, you get 2 big R, little r's to 1 little r. What about the phenotypic ratio? Well, how many different phenotypes are there? Well, so this one is red. What about these 2? Well, they do have a recessive allele, but that doesn't matter. They're also red. So there's 3 reds for every 1 white. So our phenotypic ratio ends up being 3 to 1. And so we can express these as ratios. The box is your friend. It allows you to figure out the ratios in a very easy manner. Now, did this make sense to Mendel? Let's think about what he did. He took a red thing and a white thing and he crossed them together. Everything came out red. And then he took 2 of those red things and crossed them together. And for every 3 red things, he got a white thing. He was just like, what is going on? He could not understand, but he kept doing it enough to where he saw this ratio. Now, keep in mind, you can't see the genotypic ratio. This is something that humans have sort of created, but you can see the phenotypic ratio. And for every 3 red things, he kept getting 1 white thing in this ratio. And he's like, something is going on. He was trying to figure it out. Okay? So that is what we've just done, is a really good example of what we call a monohybrid cross. Mono means one. You're looking at one trait from parents to offspring. Okay, and this is the easiest, the simplest, or least complex kind of genetics problem that we can do. And so it's really important that you take a moment to make sure you understand how this works before we get any further down the line with this genetics. Okay. Good. I think we're doing well. So some sort of permutations on what we just did. What can happen is something called incomplete dominance. I'm afraid I skipped a slide. Let's make sure I didn't. No, I'm good. What can happen is incomplete dominance. And with incomplete dominance, now unless I tell you, if I say red is dominant over white, you assume complete dominance. I would have to specify that it's incompletely dominant. But it looks like I'm doing that now. I'm specifying, I'm saying, well, actually, in this new example, red is incompletely dominant over white. And so what I want to know is, what impact on the phenotype does that have? It doesn't change the genotype. So what happens is you get a mixing of the traits but only in the heterozygote, in the heterozygous individual. So let's look. Big R is still red, right? Homozygous dominant. Little r is still white, homozygous recessive. But what about when you have one of each now? Normally, if it was complete dominance, this would be red. But guess what? Now you get a mixing of red and white, which is pink. The only thing that changes when you're dealing with incomplete dominance is the phenotype of the heterozygote. If I give you a problem and it mentions incomplete dominance, you still set the problem up the same way. You still assign alleles. You still work the problem. But then every time you come up with a heterozygote, heterozygous individual, you got to remember now it's not red, it's pink. Okay, you have a third, it gives you a third phenotype in this case. Third. Okay. Awesome. So that takes care of that. What about if you have an unknown allele and you want to know what that allele is? There's something you can do. It's called a test cross. And this is actually used all the time in breeding, particularly in plant breeding, but also perhaps in animal breeding as well. You use a test cross to determine an unknown allele. The way this works, all right, let's think about this. Suppose I said we have a white pea plant and I want to know its genotype. If I tell you I have a white pea plant, do you know with certainty what its genotype is, assuming red is dominant over white? You do, right? The only way to get white is to have 2 little r's. But what if I said I have a red pea plant? Do you know with certainty what its genotype is? You don't, right? You know what? You know it has to have 1 big R, assuming it's complete dominance. But you don't really know about that other allele, do you? But what if you want to know? What if you need to know for whatever reason? How can we figure out what that allele is? It's very simple. You take the unknown and you cross it with an individual that's homozygous recessive. Now you're going to know it's homozygous recessive because you can look at its phenotype and tell that, right? So look what we're doing. We're crossing a big R question mark with two little r's. Okay. And now when we go to figure out the gametes, we're going to get a big R or a question mark here. Here, all we're going to get is a little r. Now we draw our box. Remember the shape of the box, sometimes the gametes don't have, sometimes the parents don't make the same number of gametes. And so one thing to be aware of is the box doesn't have to be square. It can be a rectangle, as it's going to be in this case, because we don't know about the other one. So we got a big R, a little r rather. And then we got a big R for sure, and we don't know about that allele. Okay, so then we fill this in. And here's the thing, as soon as white shows up as a phenotype, then what do we know about this missing allele? It's got to be a little r, right? Soon as white shows up as a phenotype, look what you're doing. You're crossing an unknown genotype with a homozygous recessive, and then you're looking at the phenotype of the offspring to determine that missing allele. As soon as white shows up, if it does, you know that's a little r. If white never shows up, and you keep doing it over and over and over and over, and you never can get white out of it, then you would assume that that's a big R, okay? And that you're just dealing with heterozygous over and over and over and over. So a test cross is very useful. It's a way to actually figure out a missing allele, okay? Beautiful. This is great. And, you know, why would we do this? Well, you are interested because you want to know something about the organism. You want to know what that missing allele is. That's why you would do it. Okay, a couple more, and then we'll stop. This one is fascinating, and this maybe we'll go through this one, and then that'll be a good stopping point. We'll see. Environmental fluctuation in phenotype. Remember, phenotype is the physical expression, what it looks like. Okay. And interesting. It's kind of a design flaw because on my screen, this menu pops up and it covers up my slide, but that's okay. I'm dealing with it. Okay, here's the deal. In the late 1700s, this was the Industrial Revolution in Europe. And what was happening was there was a lot of excavating going on. People were digging holes to build buildings. They were digging stuff out for roads. And they started finding these fossils in the ground. And not only were they finding fossils, but they were finding these really nice progressions of fossils. So at the top of the pile, there'd be things that was obviously a horse, and they'd get a little further down. And as the further down they went, they started getting into these things which looked less and less like horses and more like things no one had ever seen on Earth before. They were finding fossils. And people were starting to ask questions. You know, back in the 1700s in Europe, the Roman Catholic Church ran the show, right? They controlled things. And people were like, well, the Bible says everything was made in its current form all at the same time. What are these bones, these fossils that we're finding in the ground? People were very confused. The church, kind of in a panic mode, decided to try to explain this. And they hired this guy named Jean-Baptiste Lamarck. And they said, you know, come up with an explanation for what's going on. And so Jean-Baptiste Lamarck, in his infinite wisdom, came up with the theory of the inheritance of acquired characteristics. He said, yeah, I can explain this. He said, a good example is a giraffe. He said, I can explain why a giraffe has a long neck. He said, you know, what happens is the mama giraffe is stretching her neck to reach the leaves higher up in the trees, and her neck gets a little bit longer. And then when she has a baby, that baby's neck is longer. And then when that baby grows up to become a mama giraffe, it stretches its neck a little more. And then when it has a baby, its neck is longer. Lamarck said, you know, in a matter of 3 or 4 generations, heck, we could go from a horse neck giraffe all the way to what we have now. Okay. What is wrong with this theory in terms of science? Can you inherit an acquired characteristic? I think the best example, maybe in Asheville, is tattoos, right? Let's say you're all tatted up and you decide to have a baby. Is your baby any more or less likely to be born with a tattoo? Of course not. Thank goodness, right? It is an environmental fluctuation in phenotype or your physical appearance. And this is not passed on. No, no, no. Right? Inheritance of acquired characteristics. You can't inherit a characteristic that you acquired while you were on the earth. It's only if it's on your genes. Okay. So Lamarck came up with this wacko theory, and people bought into it for a little while. But now in modern science, we know that this is not how things work. Another good example is that you could use would be the long neck women of Burma. If you've seen these Burmese women, this is the culture where they put the rings around the females' necks at puberty, and it actually pushes their clavicle down over time, makes them look like they have a really long neck. And this is considered to be a sign of beauty in this culture for whatever reason. That's what they're into. And I'm not going to judge them about it. It seems cruel, maybe a little bit. The men do really bizarre things too. They do the extreme earlobe stretching, and also they do the big wooden hockey puck in the lip, which is pretty bizarre. Okay, let's just say we had twins born, twin females in this culture, and to one of the females we applied these rings, and to the other we didn't. Are these women any more or less likely to have a long neck child when they give birth? Of course not, right? It doesn't work that way. It's an environmental fluctuation in their physical appearance, okay? Why do giraffes have long necks? Is it to reach leaves? Probably not. Genetic mutations occur randomly, but if there's a selective advantage to those genetic mutations, then that giraffe is more likely to survive. In this case, the one that had the longer neck was more likely to survive. But it didn't come from stretching its neck, it came from a random genetic mutation. The advantage we believe to giraffes having such long necks is actually has more to do with predation. They're prey animals. And what this does is it gives them literally a bird's eye view of the savannah where they live, and they can spot the lions and tigers that are coming to get them. And we believe this because what we've observed in nature is all the other prey animals, the shorter ones, follow the giraffes around, and giraffes are usually the first ones to give the signal that something is up. Okay. All right. We're at about 43 minutes. We'll do one last slide. And then we'll call it a day for this video anyway. Okay. What about sex determination in mammals, we might say, including us? And it turns out, and this unfortunately would be better as a superscript. I apologize for my incompetency in making the slide. I didn't do a very good job. This would be better like this. Okay. What does the S stand for? It stands for sex determination. So the gene in this case is the sex determination gene, right? And we know that if you inherit two X's, then you are biologically a female. And if you inherit an X and a Y, you are biologically a male. Now, I'm just going to preface that by saying that you can be biologically a female or a male and certainly not identify with that. And there can be biological reasons why, because genes can be turned on and off. And so, for example, you can be biologically XY, but we're exposed to high amounts of estrogen in utero. And so then you can come out feeling and in some cases even looking more female. And same thing here if you're exposed to testosterone. And so that's just one example of the fact that although we think of this as being binary, it's very much not binary. So I want to be clear about that. But for our discussions, biologically speaking, for the most part, if you have two X's, you're a female, and an X and a Y, you're a male. Okay. So there were some studies done that seemed to indicate that there were more males being born than females in the population. And, if we think about this genetically as a genetics problem, it's a monohybrid cross, right? You cross S to the X with S to the X, S to the Y. The only thing a female can donate is an X. So who determines the sex of the offspring is the male. It's the sperm, because the sperm is either carrying an X or a Y. Right? And so if we draw our Punnett square, we very quickly see that you would expect a 1-to-1 ratio of males to females. This is what we would expect. Well, some statisticians were gathering data back in the 90s, and they discovered that what they thought was statistically relevant. They found that, what they did was they gathered all the birthing data in the US for a 10-year period. This is a humongous sample size. So from the year 1990 to 2000. And what they found was that for every 100 females born, there were 109 males born. And they looked at this data and they presented it to embryologists that study these things. And the embryologists were like, whoa, this is statistically significant. There's some reason biologically why there's more males being born. And so this team of scientists got together to try to explain this. And it was, I should mention, a team of all male scientists. And their first theory was, well, you got these two sperm and one of them's carrying an X and one of them's carrying a Y. And the Y, being the male, can swim faster. That is literally what they said. They're like, these things are swimming toward the egg here. It's the Y sperm that's faster and it's more likely to get to the egg. And so they literally had sperm races. I'm not kidding. In Petri plates, they isolated X and Y sperm. They raced them across. No different, right? Didn't make any difference. And so then they struck out there. Then they said, well, actually, it's not speed, it's survival. They're like, this Y sperm is tough, man. Let me tell you, it can out-survive the X sperm in the uterus, the vagina, the uterus. And ultimately, because it lives longer, it can get to the egg. So they created these sperm torture chambers, like lots of mucus and just harsh acidic conditions. And there was no difference, made no difference at all. So they kept striking out with their theories. And so then the last thing, they actually published an article and they said, well, it actually is 50-50 to a fertilization event, but the XY zygote, the XY embryo, being the male, is more likely to be able to survive in utero. And that was their argument. So guess what happened, y'all? The next 10 years the data came out. Guess what it looks like? 110 female, to 100 male being born. Was that ever really even statistically significant? No, it was just a wobble in the data. Just like if you flip a coin 100 times, you're not going to get 50 heads and 50 tails most likely. Okay, very last thing I'll tell you about this is, let's say I had 22 daughters in a row. What are the chances my 23rd child will be a boy or a girl? Now, you might be tempted to think there's going to be a better chance of having a boy, but that's not how it works in this example. It's still 50-50. Why is that? Because these events are mutually exclusive events. They have nothing to do with each other. If I flip a coin 50 times and I get 50 heads in a row, on the 51st flip, it's still 50-50 chance of heads and tails. So don't get sucked into thinking there's some kind of ooey-wooey thing going on here. Okay? Beautiful, y'all. That was a great lecture. I hope you enjoyed it. And I'm going to sign off. We are out of time. It's been good talking with y'all. I hope you're well.